In this session, we are going to learn about photorespiration. So let us begin. Now, photorespiration is a process which occurs when the temperature is high and when there is a high intensity of light. Alright? So when the temperature is high and the intensity of light is also very high, then we will observe a process called photorespiration, especially in the case of C3 plants. Alright? So the photorespiration is specific to the C3 plants, and photorespiration is not observed in C4 plants. So if you remember, in the C3 plants we studied about the C3 cycle or the Canville cycle, where the carbon dioxide from the atmosphere was fixed into carbohydrates or sugar, right? And there were 3 steps. So in the first step, which was the carboxylation step, what happened? The primary acceptor, which was RUBP, it accepted the carbon dioxide in presence of the enzyme Rubisco, and it formed 2 molecules of 3-phosphoglyceric acid. This was the first step of the C3 cycle. Now this enzyme Rubisco, it shows 2 types of activity: carboxylase activity as well as oxygenase activity. So in the C3 cycle it showed carboxylase activity. Why? Because the concentration of carbon dioxide was more as compared to oxygen. So the enzyme Rubisco actually shows both the type of activity, carboxylase as well as oxygenase, depending on the availability or the ratio of carbon dioxide and oxygen. So if the ratio of carbon dioxide and oxygen is equal or nearly equal, then it shows affinity for the carbon dioxide and shows carboxylase activity, where it will add carbon dioxide, like what is happening in the carboxylation step of the C3 cycle, and 3-phosphoglyceric acid will be formed. But in case when the concentration of oxygen is more as compared to the carbon dioxide, if the concentration of oxygen increases more than carbon dioxide, then it shows oxygenase activity. So instead of adding carbon dioxide, it will add oxygen. And this is exactly what happens in photorespiration. So what happens in photorespiration? When the concentration of oxygen is increased, then a different product is produced. So when ribulose bisphosphate will combine with oxygen, because the concentration of oxygen is more, as in the case of photorespiration, then this Rubisco enzyme will show oxygenase activity, and it will form 1 molecule of phosphoglyceric acid plus 1 molecule of phosphoglycolate. So besides phosphoglyceric acid, 1 molecule, another molecule of phosphoglycolate is formed in case of photorespiration, and there will be no fixation of carbon dioxide. As a result, no carbohydrate or sugar will be formed, right? So no carbohydrate or sugar will be formed. So what's the use? This is a waste process, right? Because the main purpose of the C3 cycle was to produce carbohydrate, But here, when the oxygen concentration increases, then there is no fixation of carbon dioxide. Instead, phosphoglyceric acid and phosphoglycolate are produced, and the C3 cycle does not proceed further. So there will be no carbohydrate production. Alright, so this does not help the plant in any way. So this is what is photorespiration, and we studied that it occurs in the cases of high temperature and high intensity of light. So what initiates photorespiration is that whenever there is high intensity or high temperature, we studied about stomata and the functions of stomata, right? So the function of stomata is to carry out gaseous exchange. That is, it takes in carbon dioxide from the atmosphere and it releases oxygen out, which is produced during the photosynthesis. So that's the gaseous exchange, and along with that, they also carry out transpiration. The stomata, with the help of stomata, transpiration goes on, which is the loss of water from the surface of the leaves in the form of water vapor. So what happens when there is high temperature and high intensity of light? When there is high temperature and high intensity of light, then the rate of the transpiration will increase. Rate of transpiration increases, right? So more water is lost from the plant. But the plant does not want to lose more water. So what does it do? It closes its stomata. Stomata is closed. So once the stomata is closed, then the gaseous exchange stops. Now when gaseous exchange stops, what will happen? No carbon dioxide will enter inside the leaves and more oxygen will be concentrated inside the leaves. More O2 concentration in the leaves or the chloroplast, and no carbon dioxide enters the leaves. This is what happens when the stomata is closed in the case of high temperature and high intensity of light. So the carbon dioxide which was entering from the atmosphere through the stomata cannot enter now inside the leaves, and the oxygen which was being produced during the photosynthesis was released out of the leaves through the stomata, now cannot leave the stomata, and hence the oxygen concentration starts to increase inside the leaf or inside the chloroplast of the leaves. This is when the concentration of the CO2 and O2 becomes unequal, right? This is how, when the more oxygen gets concentrated inside the leaf, the concentration of oxygen increases as compared to the CO2. And when the concentration of oxygen increases, the rubisco, it starts to show the oxygenase activity. And when it shows oxygenase activity, it forms phosphoglyceric acid and phosphoglycolate, and no carbohydrate or sugar is produced. Alright, so this process, which is occurring in the case of high temperature and high intensity, where phosphoglyceric acid or phosphoglycolate is being produced because the concentration of oxygen becomes higher inside the leaves, this process is called photorespiration. Alright, photo means light and respiration means when the oxygen is utilized and CO2 is released out, right? So this is what is happening. This is taking place in the presence of light, high intensity of light, and oxygen is being utilized here because the concentration of oxygen is high. So oxygen is being utilized by Rubisco showing oxygenase activity, and CO2 is also being released out, but not in very large quantities. So this is like respiration process only. In respiration we take in oxygen and give out carbon dioxide. So this is also kind of happening inside the plant. That's why this process is called photorespiration. And this photorespiration is specific to C3 plants only. We will not see photorespiration in C4 plants. Why? We will study in a bit. But before that, let us conclude why this photorespiration is a wasteful process. So we saw that there was no carbohydrate production in photorespiration. So there is no sugar is produced, so glucose is not formed. Now, there is no production of any energy releasing compounds like ATP or NADPH. Instead, photorespiration utilizes ATP. It is not producing ATP, but it is utilizing ATP. It is utilizing the energy. And it is also releasing carbon dioxide. So we can see that it is doing more harm than good. Right? It is not doing any benefit to the plant. That is why it is a wasteful process. Photorespiration is a wasteful process for plant. And why this process happens, the reason is still not known. Reason for photorespiration is still not known. Alright, so this occurs mainly in the C3 plants. Now if we talk about the C4 plants, we studied about the C4 plants in the previous lecture, right? When we studied that, the C4 plants are those plants which grow in the places of dry and hot climate. So they are already adapted to the hot climate. And since they are present or they grow in the region with high temperature and high light intensity, so their stomata would be closed most of the times, right? And when the stomata closes, we know that photorespiration might occur. And photorespiration does not benefit the plant in any way. So these C4 plants, they have evolved with time and they have developed some adaptations inside their leaves to prevent photorespiration. So there is no photorespiration in C4 plants. And the special adaptation to prevent photorespiration is the Kranz anatomy. We studied about Kranz anatomy in detail in the previous lecture, right? What happens in the Kranz anatomy is that special type of layers of mesophyll and bundle sheath cells are present inside the vascular bundle. So what happens? The C3 cycle occurs inside the vascular bundle, inside the bundle sheath. And we learned that the bundle sheath had thick walls and they were impervious for the gaseous exchange, right? So what happens? So suppose this was vascular bundle and this was the bundle sheath. And this was the mesophyll cell. Let me use a different color. So this was the Kranz anatomy found in the C4 plants, in the leaves of the C4 plants. The upper layer is of the mesophyll cell and the inner layer is the bundle sheath cells. Now these bundle sheath cells have thick coverings and they are impervious to gaseous exchange. That means the concentration of oxygen, if it increases, as in the case of closing of stomata, that oxygen will not be able to enter the bundle sheath, and it is inside this bundle sheath layer that Rubisco is present in C4 plants. If you remember when we studied about the C4 cycle, so Rubisco is present inside the bundle sheath, right? So C3 cycle occurs in the mesophyll cell in case of C4 plants, and then C4 cycle occurs in the mesophyll cell. It starts in the mesophyll cell where the first stable product was oxaloacetic acid, right? So the oxaloacetic acid was produced inside the mesophyll cell, then it was transferred, it was converted into malate or aspartate, and then malate or aspartate was transported inside the bundle sheath. And it is inside the bundle sheath that Rubisco is present, and carbon dioxide, the concentration of carbon dioxide is more inside the bundle sheath. How? Because we studied that the malate or aspartate, it undergoes decarboxylation reaction, and carbon dioxide gas is released inside the bundle sheath. So that is how we are increasing, or the C4 plants are increasing the concentration of carbon dioxide inside the bundle sheath by carrying out decarboxylation, which occurs in the C4 cycle. And once that carbon dioxide is released, then it enters the C3 cycle inside the bundle sheath, and Rubisco is already present there. So concentration of carbon dioxide is more, and Rubisco will show carboxylase activity, and C3 cycle will occur, resulting into the formation of carbohydrate or sugar. Is it clear? If you want to study further or just to get a revision, you can watch the lecture on C4 plants and C4 cycle. Alright, so this is the reason, because of the Kranz anatomy, the C4 plants do not undergo photorespiration in spite of living in a dry and a hot climate where the temperature is high and the light intensity is very high. Why? Because there is a compartmentalization inside the leaves to prevent the oxygenation of Rubisco, to prevent the oxygenase activity of Rubisco. The bundle sheath cells have thick cell walls which does not allow gaseous exchange. So oxygen, even if the concentration of oxygen increases, it cannot pass through the bundle sheath cells. Only carbon dioxide is entering the bundle sheath cells, that too in the formation of malate or aspartate. That is not in the gaseous form, that is in the chemical form. After entering the bundle sheath, it gets converted into carbon dioxide gas because of decarboxylation, and that carbon dioxide gas increases the concentration of carbon dioxide. Because the cycle goes on continuously, concentration of carbon dioxide increases, and hence Rubisco starts showing carboxylase activity, and C3 cycle occurs and carbohydrates are produced. Alright, that is why we learned that the C4 cycles are more efficient in case of photosynthesis as compared to the C3 plants, and the productivity and yield of the C4 plants are also higher. So productivity and yield of C4 plants is higher. Also, the efficiency of photosynthesis is also higher for C4 plants. So I hope that photorespiration is clear to you. In case you have any doubts, you can write down in the comments below. And please like and share this video with your friends, and subscribe to the channel for more such content. That's all for this lecture. Thank you so much.